Good afternoon to all of you. Last time we have seen the basic part as well as the different components of ELISA. Now let us understand the different types of ELISA techniques. There are four different types of ELISA techniques: direct ELISA, indirect ELISA, sandwich ELISA, competition or inhibition ELISA. Now let us understand one by one. First is the direct method of ELISA. An antigen is immobilized in the well of ELISA plate. The antigen is then detected by antibody directly conjugated to an enzyme such as HRP. You can see this is the microtiter plate, single plate, and here the antigen is bound. The labeled antibodies are put into it. Once the substrate has been put into that well, it changes its color. So it's a direct method of ELISA technique. So advantages, what are the advantages? Direct ELISA detection is much faster than other ELISA techniques. The assay is also less prone to errors since fewer reagents and steps. Now, of course, very few reagents have been needed. Best for analyzing the immune response to an antigen. Disadvantages: antigen immobilization is not specific, so that is one of the disadvantage. Less flexible and no signal amplification, which is helpful for reducing the sensitivity of this particular kind of method. Indirect ELISA. Antigen is observed to well in an ELISA plate. Detection is a two-step process. First, an unlabeled primary antibody binds to specific antigen, and second, an enzyme conjugated secondary antibody that is direct against the host species of the primary antibody. So here you can see in this diagram, the inside the microtiter plate, you can see here the antigen is being bound, and once the antigen is bound in here, the antigen has been placed in here, the primary antibodies are being put. Then this antibodies which are of secondary type are being put, and those bind to this antigen antibody complex, and once you put the substrate, then it converts it into product. Advantages: economical, highly sensitive, greater flexibility, best for determining total antibody concentration in the sample. So that has been extensively utilized for not only for detection of HIV but detection of various different other components of proteins as well. Disadvantages: possibility of background noise, longer procedure in the direct. Yes, since it needs the indirect way, therefore it will take a long duration of time. Additional incubation step for secondary antibodies will be needed. Sandwich ELISA. Sandwich ELISA requires the use of matched pairs. So it requires the matched antibody pairs, that is capture and detection antibodies. Each antibody is therefore specific for a different and non-overlapping region of the epitope of the antigen. The capture antibody binds the antigen that can then be detected in direct ELISA or indirect ELISA configuration. So here, instead of an antigen this time, the antibody has been fixed. These antibodies can be taken from the blood of the particular patient. Once it has been centrifuged, the serum will contain the antibodies, and then these antibodies can be fixed inside the well. Once you pour the antigen, if it has been specific antigen, then it will bind to that antibody. After binding it, we now can put the antibody, which is another antibody which is specific for enzyme, and this will bind to the specific antigen. You see the binding here, and the antigen is said to be sandwiched between two antibodies, and then therefore this method is called as sandwich ELISA. So what are the advantages? Highly sensitive, 2 to 5 times more sensitive than direct or indirect ELISA. High specificity, 2 antibodies are involved in capture and detection. Flexibility, both direct and indirect detection can be used. Best for analysis of complex samples, since the antigen does not need to be purified prior to the measurement. Disadvantages: antibody optimization can be difficult. Cross reactivity may occur between the capture and the detection antibody. So this is one of the major disadvantages, but this is one of the best techniques that has extensively been utilized. Quantitative inhibition ELISA. The competition or inhibition ELISA is also known as the blocking ELISA, is the most complex of all the ELISA method, used to measure the concentration of an antigen or antibody in a sample by detecting interference in an expected signal output. Sample antigen or antibody competes with the reference for binding the limit amount of labeled antibodies or antigen respectively. We have seen a part in antigen antibody reactions which is called as competitive reactions. So this is the same kind of thing. The higher the sample antigen concentration, the weaker the output signal, indicating that the signal output irreversibly correlates with the amount of antigen present in the sample. See here, prior to this test, labeled antibodies, label antigen patient sample, and ultimately the test is done. Advantages: no sample processing is required, and crude and impure sample can also be put in. More robust, less sensitive to sample dilutions and sample matrix effect than sandwich ELISA. More consistent, less variability between duplicate samples and assays. Maximum flexibility rather than direct, indirect, or sandwich ELISA. Commonly used when only one antibody is available for the antigen of interest. It is also suitable for detecting small antigens that cannot be bound by two different antibodies, such as sandwich ELISA technique. See here, this is direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So one single slide will decide all the 4 different types of techniques that we have learned today. Now, the important part: applications used for determining serum antibody concentration. Monoclonal antibody screening can be extensively done with the help of ELISA. For virus tests such as HIV, West Nile virus, NDV, for home pregnancy tests. In food industry for detecting potential food allergens, milk, peanuts, almonds, eggs, walnuts, all can be detected for the adulteration or allergens. Disease diagnosis for HIV, bird flu, common colds, cholera, STD as well can be detected with the help of this particular kind of technique. Detection of enterotoxins of E. coli in feces can also be done with the help of ELISA technique, and the drug toxicity studies can also be done with the help of this particular kind of technique. Correct. So today we have end up this part of ELISA. Why I have taken this lecture of 3 different parts? The reason behind it is that this is the most extensive and excellent technique in immunology that can be useful for detection of antigen antibody reactions. Correct? So that's all from today.